I want to get to some of the recent research you've done, and some of the background here that I think is relevant is we've talked about the microbiome already. I've done a number of episodes about the gut microbiome, so I'm not sure how much background we need to do there, but obviously we have a gut microbiome that's very important in different ways, and the immune system ties into this for the reasons we discussed. The immune system is there to sort of monitor and keep the gut microbes where they're supposed to be, and to determine how some of that happens. One of the nutrients that I know that you've looked at that's going to be relevant here is vitamin D. And I want to have a fairly general discussion before we get into the findings. Vitamin D is quite a famous nutrient. Everyone's familiar with it to some extent. Most of us, my understanding is most of us are apparently vitamin D deficient most of the time these days for reasons that aren't entirely clear to me. And vitamin D is very important for a number of processes in the body. And I believe it's important for immune system function generally, although I don't really know much about that. So, what is vitamin D doing that's relevant to the immune system?
So I should say that our recent work that has led us to vitamin D was one of those serendipitous discoveries in
Mm,
science. We were not interested in vitamin D whatsoever, and we had never really worked on vitamins or nutrients or even the microbiome,
hmm,
uh, and for various reasons, which actually have to do with the actin cytoskeleton, which we were studying, these sort of signals of cell death recognized by dendritic cells, which we discussed earlier. For we were looking for mechanisms by which the immune system might modulate its response to uh, this actin skeleton of cells, and we stumbled upon a protein that was modulating vitamin D availability. So there is a very, very long history of vitamin D and everything. Vitamin D immune system, vitamin D in cancer, and vitamin D in health in general. And I don't really want to get into because a lot of those studies are quite controversial, and some you will find evidence for all sorts of different ideas. The only thing that I can say with respect to vitamin D is our own work, where we serendipitously found that vitamin D was increasing the resistance of experimental mice to challenge with cancer. And we thought that this was something rather odd. That's not what we were studying. But once we found it, we decided to get to the bottom of it. And basically, what we ended up finding is that a very convoluted, if you will, mechanism by which what vitamin D is doing is modulating the microbiome, the composition of the microbiome, we think, allowing for some bacterial species to thrive or do a little bit better than others. Uh, and in turn, those bacteria were instructing our immune system to be more responsive to the presence of cancer.
hmm.
Now, that raises a lot of interesting questions, some of which relate to what we were already talking about. Now, we said that the immune system does not ignore these commensals and, in fact, actively keeps them at bay, albeit with a atypical response that does not cause elimination. But it turns out that our immune system is also being conditioned by those bugs. So there is a sort of dialogue going on where things that made by those commensal bacteria in our guts and perhaps commensal fungi or even commensal viruses are putting our immune system on different states of alert, and that can have an impact on immune responses elsewhere. In this particular case, in immune responses to cancers that are nowhere near the gut. Uh, and what vitamin D is doing is impacting this dialogue indirectly by effectively allowing some bacteria to thrive that make some more of perhaps of the compounds that put the system on a cancer alert state. Um, uh, and the way that vitamin D is doing that is actually by acting on these epithelial cells of the gut that we were talking about, the ones that form the barrier. And those epithelial cells of the gut are not just passive components of the dialogue between the commensal bacteria and the host. They are active participants because they can make little molecules that get incorporated into that mucus and that help keep the bacteria at bay and that may favor some bacteria versus others. So, in a nutshell, what seems to be happening is vitamin D acts on epithelial cells. Epithelial cells make something or stop making something, we don't quite know, that helps change the composition of the gut bacteria. This is all in mice, I should say. That altered composition now changes the dialogue with the immune system. The immune system, in response to whatever signals are made by whichever bacteria thrive when vitamin D has acted on the epithelial cells, that immune system is now more responsive when we challenge mice with a cancer.
Hmm.
So, just to be clear, this has all been done in mice, and to what extent this is all applicable to humans, we don't know. But it did lead us to sort of ask a little bit about vitamin D and human cancer. And we were lucky to actually be able to collaborate with some colleagues in Denmark who had access to a very, very large database of health records of the Danish population. You alluded to the fact that vitamin D can be limiting for many people, and because it can be made by sun exposure, uh, populations that live at more northern latitudes like Denmark tend to experience more of a vitamin D winter. So vitamin D may be more limiting for those populations. And what we were able to do is stratify people from the Danish population into those that had high, low, or deficient vitamin D based on a single blood test. So
Hmm.
there's caveats to this because we don't know what they did after that blood test. They could have taken supplements to correct any deficiency that could have been detected. But nevertheless, we had 1.5 million people. We could divide them into these three categories. And what we found is looking at their health records is that those that had once been diagnosed as vitamin D deficient over the following decade had a higher risk of developing a number of cancers compared to those that were not vitamin D deficient.
Hmm.
Now, that's the extent, really, of our human correlate. It's just a correlation in humans. In mice, we can prove all of these connections that I've just told you about. In humans, we can only observe and correlate. So we do not know to what extent we're dealing with the same issue of somehow the vitamin D affecting the microbiome also in humans.
Hmm. So on the basic biology side, the stuff that you did in mice, so it sounds like maybe a way to think about this is the immune system is constantly monitoring the gut. It's constantly in dialogue with all of the bacteria in the gut microbiome. And the specific composition of bacteria in the gut microbiome, how many are there, which species are the most prevalent, this is going to affect the general behavior of cells in the immune system. So, by analogy, I might imagine like in my own life, I might have a conversation with someone and this has a calming effect on me. I might talk to somebody else and they provoke anxiety. And, the state they put me in through my dialogue with them is going to affect other aspects of my life. I might be more likely to, trip and fall or be a little bit more cautious depending on who I was just speaking to. And so the composition of the gut microbiome is going to sort of determine the readiness or the propensity of the immune system to be on high alert or lower alert for cancer cells. And so by modulating the composition of the microbiome, the vitamin D status of mice, at least, indirectly influences how readily the immune system is prepared to detect cancer, basically.
That's a very good analogy, and I think it's a pretty apt analogy. The only thing I would add to it is that we don't even know if it's merely the composition, i.e., the people that you encounter, or maybe the clothes that they are wearing. So maybe the
Mm.
same person wearing a red shirt will give you more anxiety than that person wearing a blue shirt. And so it could be as subtle as that, not just the people who are there, but what they are wearing or some other aspect of their appearance.
Hmm. Interesting. And then, we suspect this could be at work in humans, but it hasn't been proven yet. But you've at least shown that there's a correlation between the vitamin D status of certain people in certain parts of the world and cancer rates.
Yes, and the only other piece of human data that we were able to get at is to sort of indirectly measure the genes that are induced by vitamin D in their cancers, where they had a cancer biopsy, and try and correlate that with outcome and responses to immunotherapies. And again, by that sort of somewhat indirect measurement of their vitamin D status, again, we came to the conclusion that it was having a higher vitamin D status led to better outcomes.
Mhm. And are these for all of this stuff, are there specific forms of cancer that you guys were looking at, or is it a general phenomenon?
It was actually quite general looking at these human populations. There were a number of cancers for which we didn't see any correlation one way or another. Where we did see a correlation across a number of different cancers, it was always that vitamin D um, insufficiency correlated with worse outcomes, i.e., increased incidence of cancer. The only cancer for which we saw the inverse was skin cancer.
Hmm.
But skin cancer is a confounder, of course, because since
I see. I
exposure
see.
to light gives you vitamin D, but it also is a risk factor for cancer. We suspect that could explain why we saw an inverse correlation there. And in that case, the vitamin D have been a indication or a marker of sun exposure more than anything else. So we actually excluded those data
Mhm.
from this type of analysis.
And do we know much about what exactly vitamin D is doing within cells mechanistically to elicit this whole pattern?
No, we don't. I mean, using mouse genetics, we found that it was acting on gut epithelial cells. That came
Hmm.
as a little bit of a surprise. And so vitamin D acts on a receptor called vitamin D receptor, not surprisingly. And what we basically found using mouse genetics is that if we took vitamin D receptor away from epithelial cells, then the whole benefit of vitamin D went away. So, what exactly it does there, we don't know, and how exactly whatever it does then modulates this composition of the microbiota, we don't know. These are all areas that we are sort of exploring at the moment.
Hmm. So vitamin D is itself a signaling molecule, and its receptor is it an extracellular receptor that's in the cell membrane?
No, it's not. So vitamin D receptor is part of this somewhat small family of receptors that lives inside the cell, the nuclear receptors.
Mm,
The vitamin D actually diffuses into the cell and binds to the receptor in the cell.
I see. So that actually makes sense. Vitamin D is fat soluble, so it can actually get into the cell.
Exactly. Yeah.
Interesting.
I mean the other parameter that we've sort of identified here is that because vitamin D is fat soluble, it needs a carrier protein in the blood. That protein is called GC globulin, and it was actually when we looked at mice deficient in GC globulin that we found this whole phenomenon, which was rather unusual. And but it is interesting because it turns out that the GC protein or the gene that encodes the GC protein is very variable amongst us humans. So it has lots of different variants. And there are studies that associate certain variants with vitamin D deficiency.
Hmm.
So actually, it turns out that it's not only about exposure to light, which can allow our skin to make vitamin D or active vitamin D. It's not just about diet, which is another source of vitamin D, but then your own ability to transport vitamin D and the genetics that you have in terms of the vitamin D carrier might all come into play. So if we're then thinking about human disease or human health, I think it's going to be a very complicated thing to dissect because you have to take all of these different factors into account.
Wow. Yeah. So it's not just about vitamin D synthesis or vitamin D supplementation, your body's ability to actually transport the vitamin D through the blood to where it needs to go is going to be a major player here. And there's a lot of genetic variability in how effectively our bodies do that.
Correct.
What are some of the other things you're working on in the lab today in terms of how the innate immune response works and anything related to this work?
Yeah, so I mean, we are continuing the work to try and understand how vitamin D modulates cancer immunity. But I mean, a lot of the core work at the moment in my lab has to do with understanding dendritic cells. I mean, we talked about dendritic cells as if they were one cell type. They are one white blood cell type, but they come in multiple flavors or subtypes, and no one fully understands whether they all do the same thing or slightly different things. So that's an area that we sort of focus on. Perhaps the area that we're putting a lot of effort into is this understanding of cell death and how that translates into cancer immunity. I mean, that goes back to the very beginning of our discussion when we were uh, pointing out we were asking how does the immune system detect the presence of cancer cells? And I was saying one of the ways is by detecting death of cancer cells. Uh, I think this was something that had been postulated before, but hadn't really been shown properly. I think we've now, over the years, been able to show that. And we tried to then understand whether, having shown it, can we now leverage that to make better immunotherapies? So actually, a couple of years ago, I spun out a biotech company called Adendra Therapeutics that is effectively trying to make cancer cell death more immunogenic, if you will, i.e., use our knowledge of how dendritic cells handle dead cells, dead cancer cells, to try and see if we can make the system more efficient, and even if we can persuade other cells besides dendritic cells to do the same thing, thereby increasing the number of sentinels that might be around in a cancer to alert the T cells against it. So we are doing quite a lot of work trying to develop biological agents that we believe might help with this. And we'll have to wait and see if they turn out to be useful. But that is a sort of uh, example of how what is effectively a purely academic project to try and understand how things work can nevertheless be leveraged potentially to develop classes of therapeutics that weren't really envisaged before. So that's an area that's keeping us quite busy at the moment. And the final area that's keeping us quite busy at the moment is understanding what the dendritic cell does when it takes up these bits of dead cells from a cancer, for example. Um, And we discussed how these dendritic cells put up bits of bacteria and present them to T cells. Well, they do exactly the same for cancer cells. They take up bits of the cancer cells or of the dying cancer or dead cancer cells and present those bits thereof to T cells. Now, how cell biologically that happens turns out to be quite fascinating. It turns out that the dendritic cells take up these bits of dead cells into little vesicles that are inside them. And then they rupture those vesicles and disgorge the contents into the cytoplasm, which is something very unusual. So these are what are called endosomes or phagosomes. And dendritic cells have a specialized mechanism where, in response to detection of these dead cell debris, the detection of the actin cytoskeleton, they actually actively signal to disrupt these structures and disgorge these contents for easier handling of the material for presentation to T cells. And that's a cell biological process that, to my knowledge, hadn't really been described before. We described it about 3 years ago, and so we are actively trying to understand how that is actually regulated. Again, with the idea that not only it tells us something about biology and about cell biology in this case, but perhaps we can exploit it to make it more efficient.